Hello, am I audible? Please write in comment section. Hello, if I am audible or not. Okay. Good evening, everyone. Hi, this is Vaishali Kamdar from Spark Academy, and today we will be discussing something that is one more important topic of your second year, that is biotechnology principles and processes. Principles and processes involved in biotechnology. So it will be very easier for you to remember all these techniques after the session. Okay, so don't go anywhere. Stay tuned. Those who have not joined, ask them to join because it will be a very useful session for your NEET exam. It's only 1 month remaining. Okay. So stay tuned. Don't go anywhere in between, please, because this will secure your 16 marks. Okay, so hi, this is Vaishali Kamdar from Spark Academy. Before starting this session, I would insist all of you to join us on any of these links. You can join us on YouTube, Instagram, Telegram, any of this link. Okay, or else you can download the app, Spark Academy app from Play Store, and then join any of the course which are offering right now, that is for NEET, MSET and JEE mains, okay, JEE. So that after that, after joining the course, you will be having free study materials, free previous year questions solution, free videos, exclusive live class are continuously going on. This live class, you will be access to all you will be free access to all this live classes. Okay, so what I mean, this is how live classes are going on, and you can join the live class at that particular time, and you can ask your doubts in chat. We will be solving all your doubts at that time only. Okay, and then you can check this video afterwards also, whenever you require, whenever you have any doubt while reading, while referring, you can check this video afterwards also. You can follow us, you can start from the starting beginning of the chapter, beginning of the year, and you can check out all the videos afterwards also. So it's, isn't it nice? Yes. So, what are you all waiting for? Download the app, Spark Academy app, and then join any of the courses, and you can have all this access. We are assigning assignments on regular basis. Okay, so you'll be access. I mean, we are ensuring that you are doing a very good practice if you follow all these assignments. If you submit all these assignments on time, your practice will be first class. So this is how these all courses are going on right now. Okay, and right now it's on 50% discount. Right now it's on 50% discount, so uh, you can just add the Spark 50 code, and you will be having 50% discount. Or else you can call us on these many numbers which are given for admissions. Okay. Now, biotechnology principles and processes. Okay, in the first topic of the biotechnology unit, in this chapter, I mean, from this topic, mostly 4 questions are asked. So 4 questions gives you 16 marks. Okay, so it's a very important chapter. Okay, very technical, various techniques we need to discuss here of a recombinant DNA technology in this chapter. Okay, so don't go anywhere, stay tuned. And those who have not joined, ask them to join, because it will be very useful session for you all. to secure all the to secure the 16 marks. After completion of this chapter, after completion of the session, all your 16 marks will be secured. Okay, so 4.44 percentage weightage in terms of Neet of this chapter. So it's very important chapter. So don't go anywhere. So we are starting biotechnology now. The biotechnology term was coined by Carl Ureki, and it was a technique. Biotechnology is a technique using living organism or their cellular components or enzymes to produce useful products and processes for men. Okay, we are going to produce you. We will be going to produce useful products by using some living organism. Okay, so according to European Federation of Biotechnology, this is very important definition. Okay, very important definition of biotechnology. One second, I'll change the color of pen. So very important definition of biotechnology, that is biotechnology is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for the products and service. Development of biotechnology can be studied under two phases. Two phases include old and traditional biotechnology, and one is modern biotechnology. Now we are going to discuss about modern biotechnology. So among many, two core techniques that enable birth of modern biotechnology, they are genetic engineering. So we are engineering here. Here we are going to engineer genes. So in this technique, chemistry of genetic material, DNA and RNA is altered and introduce these to the host organism, and thus alter the phenotype. Okay, whatever characteristics you want. For example, you want brown colors or what's a cat eyes in your baby. Okay, that characteristic eyes of the genes responsible for eye of your baby, eye of coming baby, that gene you can modify it. Okay, so when it is in genotype, you are modifying, you are altering genotype. So why you are altering genotype? To get your desirable phenotype. What is your desirable phenotype? Color of eye. Okay, you want cat eyes. You want cat eyes in that. So these type of genetic engineering, recombinant DNA technology, this is called genetic engineering. Okay, that we can do, and we can introduce this altered type of gene which have our desired phenotype to produce. Okay, in host cell. This is called genetic engineering. Next, maintenance of microbial contamination free surroundings in a chemical engineering. So it helps in the growth of only the desired microorganism, eukaryotic in large quantities for the manufacture of the biotechnological products such as antibiotics, vaccines, enzymes, medicines, hormones, etc. Okay, so we are enabling, all the microbial contaminants, we are not allowing them to grow. We are only allowing those organisms which are of our use. Okay, they can produce. So we are modifying in that way, that way with the help of genetic engineering. All the environment we can modify. Okay, our surroundings can be modified. Next, genetically modified organisms and transgenic organisms. We have already discussed GMOs in detail in previous class, where we have discussed biotechnology technical part. Okay, GMOs related agriculture and all these things we have discussed in last previous lecture. You can find out that in YouTube. Okay, I have discussed that in previous class before a month. Okay, so GMOs, they are known as genetically modified organisms. So GMOs can be developed by following three basic steps. Identification of DNA with desirable gene. Okay, here first you should identify that particular DNA which is yours desirable. For example, I have taken one example, that is cat eyes, gene for the cat eyes. Okay, gene for the what say brown eyes, color of eyes, that is brown eye. Okay, what is my desirable character? Gene for the brown eye. Okay, so first I need to recognize, I need to identify that gene which is responsible for color of eye. Okay, that is my desirable gene. Then introduction of identified DNA into the host cell. This brown eye gene responsible for the brown color of eye, that is I am going to introducing in the host cell. Okay, then maintenance of introduced DNA in a host cell and transfer of DNA to the progeny. So how they are going to maintain? So if it is going to be transferred and this DNA is transferred to the progeny, then and then it will going to maintain. Okay, if we are talking about this brown eye, here we are talking about humans, but most probably in GMOs, I mean here we are talking about microorganisms. So then and then if they are transferring to progeny, then and then your particular gene is replicating. Okay, am I clear everyone? Can I have some quick comments if it is okay, if it is understood? Then what are GMOs? GMOs are genetically modified organisms are created through biotechnology, also called genetic engineering. DNA is altered artificially in lab with genes for other plants, animals, viruses, and bacteria to produce foreign compound. Okay, GMOs cannot occur in nature. Genetically modified, we are going to modify that particular gene in lab. So they cannot be natural. Okay, they cannot be present, occur in nature. Okay, these are some few examples to you. Photographs of GMOs, genetically modified fruits. Organism which is organism here? Fruits. Now recombinant DNA technology. Stanley Cohen and Herbert Boyer. Cohen and Boyer in 1972 first constructed recombinant DNA. They first constructed this recombinant DNA. They isolated a piece of DNA from a plasmid. Okay, what is plasmid? Can I have some quick comments, everyone? Those who have joined, what are called plasmids? They are chromosomal DNA or they are extra chromosomal DNA. What is the meaning of plasmid? Plasmid. They are circular or linear. Write in your comment section, everyone. Stay tuned, don't go anywhere. Take active participation in the class. So plasmid is circular. Yes, it's circular DNA. Circular DNA, which is also known as extra chromosomal DNA. So they isolated a piece of DNA from plasmid carrying antibiotic resistance gene of the bacterium Salmonella typhimurium and fuse it with the plasmid of E. coli. Okay, what antibiotic resistance gene? Actually, it was present in the plasmid of Salmonella typhimurium. Species name, that is Salmonella typhimurium, and they wanted it to transmit in, it is going to fuse with the plasmid of E. coli. Plasmid is used as a vector. Mostly plasmids and phages, bacteriophages, they are used as vector because they are irrespective. They are not in control of the DNA, chromosomal DNA. Okay, they can replicate on their own. Even bacteriophage, even the DNA of any bacteriophage can replicate inside the bacterial cell without any instruction from chromosomal DNA. Okay, so plasmid is used as vector to uh, carry an align piece of DNA into the host organism. The linking of these two is performed by DNA ligase. So this is also known as molecular glue. One is cutting, now one is joining. So DNA ligating. Ligase words come from the ligating, that means glue. Gluing two DNAs. Gluing of our desired piece of DNA to the our desired organism, plasmid of our desired organism. So linking of these two is performed by DNA ligase, resulting in recombinant DNA. Now it is transferred into E. coli, where it can replicate in presence of new host DNA polymerase enzyme and form multiple copies that represent gene cloning. So now what you have done? You have cloned a gene in E. coli. So now what are all tools you need for the recombinant DNA technology? For this rDNA technology, you will require enzymes, few enzymes to cut down and to join, okay, and cloning vector and competent host. That means a host should acquire that gene and it can allow that gene to multiply. Okay, the host should be competent here. So we will discuss one by one what all enzymes you will require for our DNA technology. First, enzymes for recombinant DNA technology. So enzymes you require first, it is restriction enzymes. What is the meaning of restriction enzymes? Enzymes which break down DNA. There are mainly there are three types of restriction enzymes: exonuclease, endonuclease, and restriction endonuclease. One which is, see, this is exonuclease. One second. See, this is example of exonuclease, where it is cutting the DNA from backside. Okay, it is cutting, or it is exonuclease separate nucleotides from the terminal ends, from the terminal ends. That those type of restriction enzymes are known as exonuclease. Endonuclease, which cuts from the inside. Okay, they form cuts at the specific position within the DNA, at the specific position in DNA. So inside. And restriction endonuclease, they cut the DNA strand at the specific base sequence in palindromic sequence site, thus the function as the molecular scissors. One is doing activity of scissors, one is, now next enzyme will be gluing, which will be having activity of glue. Okay, now see what we are doing in rDNA technology. In this genetic engineering, we are crafting. Okay, when you want to do craft, when you want to perform any craft, make anything from paper, you need to cut paper pieces. So first you will need scissors. Okay, so which are known as molecular scissors, restriction endonuclease. Then you need to glue it properly. Okay, so next it is gluing. We'll come to that point. First we'll discuss about restriction endonuclease in detail. So these are three scientists who discovered these enzymes. Restriction endonuclease first inspect in the DNA sequence, it recognizes its specific sequence, then binds at that sequence. Okay, that sequence is known as palindromic sequence and cut at that point. What it will cut? It will cut down sugar phosphate backbone. Okay, sequence which is recognized, it is known as palindromic sequence. Palindromic sequence is also, it is the sequence when you read from both the sides, forward or backward, it will be having same words. It will form say same word. Okay. So restriction enzyme that after cutting down, it leaves, if it will leave single stranded unpaired cut ends, that means it is leaving behind sticky ends. These ends, if it is leaving single stranded ends, then these ends are known as sticky ends. So it will facilitate the activity of ligazing. Okay. Then if two strands are separated, then only you can ligate it. Okay. So next, first restriction endonuclease that was isolated, that is HIND2. It was isolated from Haemophilus influenza RD. That's why it is known as HIND2. Okay. It creates blunt ends. Now what is the difference between sticky ends and blunt ends? Look here. Here, sticky ends, that means, one second, blunt end, that means these are blunt ends. Okay, if it is cutting from the same target point, it is cutting at the both the side at the same point, then it will generate this type of blunt end. This is called blunt end, and this is called sticky ends, where one single-stranded DNA is generated. That's why it is called sticky ends or cohesive ends. Okay. Now next, it is genetic glue or molecular gluing technique. First, scissors, molecular scissors, that was a restriction endonuclease. Now it's ligase. So ligate, it will perform as gluing activities. It is ligating two strands. Then alkaline phosphatase, it helps to remove phosphate group from the end. Okay, whatever phosphate group remaining at the 5' end, it will remove it. Okay, leaving a free 5' hydroxyl group. It can be isolated from calf intestine of a calf intestine or bacteria. Then DNA polymerase. DNA polymerase. It is employed to polymerize replication of DNA on DNA template or complementary DNA. So from complementary DNA, see, this is a polymerase which is making a DNA from a complementary strand. It's catalyzed from 5' to 3' and 3' to 5' exonucleotide degradation of DNA. DNA polymerase firstly investigated by Kornberg in E. coli, and now it is known as DNA polymerase 1. Other two enzymes, DNA polymerase 2 and 3, are also isolated. Among this, 3 is more active. It is more active. This is example of DNA polymerase 3. Okay, it produces a parallel strand in the presence of ATP on DNA template. Second thing, what we require in our DNA technology, first it was enzymes. Now second it is, first it were enzymes. Now second it is cloning vector. One of the most important cloning vector, it was PBR322. A plasmid, name of that plasmid is PBR322. Okay, other cloning vectors, plasmid, bacteriophage, cosmids, phagemids, yeast artificial chromosomes, bacterial artificial chromosomes, transposons, these all are examples of cloning vector which are used. Even shuttle vector, which we make in our own desirable thing, so that is also known as shuttle vector. So plasmid, first, it is extrachromosomal, self-replicating, usually circular, double-stranded DNA molecule found in many bacteria and some yeast. They may occur as in one or two copies or in multiple copies inside the host organism. The ideal plasmid here, it is PBR322. PBr322 as a vector. So P denotes here for plasmids, BR stands for Bolivar and Rodriguez, who constructed this plasmid, and 322 is the number given to distinguish this plasmid from other developed in the same laboratory. Okay, so that's why it is PBR. BR stands for Bolivar and Rodriguez. So what is here in PBR 322? Structure of PBR 322. There is presence of ori site, from where it is also known as origin of replication, from where replication starts. Then it is having a marker gene which is known as antibiotic resistance genes, that is ampicillin resistant gene. Okay, ampicillin and tetracycline resistance gene are present at PBR322. If this plasmid is going to transfer in a host cell, it will get resistance against ampicillin and tetracycline both. Unique recognition site for the restriction endonuclease. It has a variety of unique recognition sites for restriction endonuclease. So two unique sites, that is PST1 and PVU1, located inside the ampicillin resistance gene, which all characteristics we need in a cloning vector. For a successful, powerful cloning vector, we need these many characteristics in a cloning vector. First, it should have origin site. Okay, so for example, now we are designing our cloning vector, our desirable cloning vector, through which we are going to transfer our desired gene. Okay, so it consists, one is origin, okay, second a marker gene, then third a recognition site for cloning. So recognition site, okay, selectable marker gene. So how can you select if you are getting cloning or not? How can you select that your transformation has occurred or not? So it should have any marker gene. Okay, so vector also requires to identify and eliminate non-transformant and selectively permit the growth of the transformant only. So genes encoding resistance to the antibiotics such as tetracycline, ampicillin, kanamycin or chloramphenicol are useful in selectable markers for E. coli. The next, recognition site. Vector bears one unique restriction endonuclease recognition site, from which it will cut, and then we will join our desirable gene. So same restriction site should be present in both the DNA, which enables foreign DNA to be inserted into the vector during the formation of recombinant DNA molecule. Then color reaction. Okay, it should give some color reaction to select for the selection purpose. So due to inactivation of antibiotics, selection of recombinant DNA recombinants becomes burdensome process, because it is required stimulation of planting on the two plates having different antibiotics. Thus, alternative selectable marker is developed to see. First, it was used, that is antibiotic resistance genes only for selection purpose. Now, one more color reaction gene it is also introducing in the cloning vector, that is alternative selectable markers is developed to differentiate recombinants and non-recombinants on the basis of ability to produce color in the presence of chromogenic substances. Chromogenic, that means related to colored substances. So for example, if your desirable gene, it was color of eye, brown color eye. Okay, so that gene, if that gene is transferred along with the other genes present in the vector, okay, so if vector is containing any selectable marker which gives color reaction in a particular media, Okay, now it can give color product, color colonies, so that you can select those colonies, only those colonies from the master plate. Okay, here what you need to perform? Replica plating. Replica plating. We will discuss that. Okay, so those color products, color colonies, you can select using replica plating. See, this is how you can do replica plating. See, there is one type of one minute. Here you can use replica block, just like a stamp, a bigger stamp you can say. This is called replica block. In this, it is covered with some velvet material. Okay, so what you need to do, you just touch this replica block on this master plate and transfer it to your plate number 1. Okay, so this is your master plate. So master plate will be having all type of colonies, white colonies, red colonies, green colonies, all type of colonies, colonies of transformants and colonies of non-transformant also. Okay, so here the blue transformant, not having they are not having inserted DNA. Okay, and this are having recombinant plasmid. So you can select that again from the master plate. If they are growing in tetracycline, they are growing in ampicillin, then you can select it. Okay, so selection of recombinant DNA, recombinants by non-transform, non-transform cannot grow in presence of ampicillin and tetracycline in your medium. For example, you are performing replica plating. Okay, from this master plate, you are inoculating into 2 plates. One plate is containing ampicillin and one plate is containing tetracycline. So, those who are not transformed, they cannot grow in these plates. Okay, only transformed colonies can grow in ampicillin and tetracycline containing medium. Now transformed with non-recombinant. See, this ampicillin and tetracycline gene is not of our desired gene. Okay, so how can we recognize this if it is containing our desired gene? So an unaltered vector can grow in both ampicillin and tetracycline containing medium. Okay, the transformed with recombinant vector can carrying one gene of interest. This transformed recombinant can grow only in ampicillin medium and cannot grow on tetracycline medium because your desired gene you have inserted in between tetracycline gene. So that's how you can select that particular recombinant transformed from master plate. So this is your green color, this green color colony, it is what of our desired recombinant vector. Am I clear, everyone? So then vectors for cloning gene in plants and animals. You all know about Bacterium tumefaciens, that is tDNA. That's why tDNA is transferred because transformed and GMOs are created. Now let's start with the question answer session. First question answer session. That is first question. It is. Come on, I want quick answers from your side. Take active participation in this class. Write in your comment section. First, you need to write it with question number. So 1A, B, C, D like that. Okay. A genetic clone is what? What is true for a genetic clone? A plant produced by asexual means, hybrid produced by sexual means, homozygous plant produced by sexual means, and heterozygous plant produced by sexual means. Which of this is correct answer? Genetic clone. Genetic clone. Word here, that is genetic clone. Second. Correct answer is a plant produced by asexual means. Then PCR, polymerase chain reaction method, is useful for amplification of DNA for forming billions of copies of itself. Monoclonal antibody production, hybridoma production, or all of above. PCR used for the amplification of DNA for forming billions of copies of DNA. Polymerase chain reaction. Correct answer, it's 2A. Which structure involved in genetic engineering? Which structure that is involved in genetic engineering? Correct answer is structure. I am talking about structure which is involved in genetic engineering. Codon. Sorry, it's plasmid which is involved in genetic engineering. Plasmid which is involved in genetic engineering. Genetic engineering aims at what? Destroying wild genes, preserving defective genes, curing human diseases by introducing a new gene, hemophilia, or all of above. Correct answer is 4C. Ti plasmid used in genetic engineering has been modified by Ti plasmid has been modified by yes, it has been modified by deleting tumor forming genes. Deleting tumor forming genes. Which of the following technique is used for the separation of DNA fragments? Separation of DNA fragments. Yes, it is gel electrophoresis. Which of the following is the tool for recombinant DNA technology? We have discussed tools for the recombinant DNA technology. Cloning vectors like enzyme restriction enzymes, all of above. Correct answer, we need all these things, all these three things. Which of these is not correctly matched? Gene gun, bioelastic gun, plasmid extrachromosomal DNA. Not, you need to focus on not word. DNA ligase, biological scissors. DNA ligase is biological glue. So this is not correct, not correctly matched. So this is correct answer. Conversion of sugar into alcohol during fermentation is due to direct action of what? Direct action of what? In fermentation. Ninth answer is ENZYMES. A single strand of nucleic acid tagged with a radioactive molecule is it is known as probe. Okay. Now third thing which we require for our DNA technology, third tool, that is competent host cell. Competent host cell. It is essential for transforming with recombinant DNA. Since DNA is hydrophilic molecule, it cannot pass through the membranes. So the bacterial cells must be made capable of take up of DNA. This is done by treating them with a specific concentration of divalent cations such as calcium. This question could be asked. It is very important point. Calcium, it is treated with the calcium and increase in efficiency with which DNA enters the bacterium through the pores. Okay, this is how we can make cells competent. Okay, Then gene transfer. Gene transfer can be done by microinjection. Gene guns can be used. Gene guns are also known as particle gun or biolistic guns. Direct DNA injections can be given, and electroporation. Electroporation. First, microinjection. This is how microinjection needle containing foreign DNA. It is given directly to the nucleus. Okay. Introduction of foreign DNA in host cell with the microinjection needle. Then gene guns are used. These are pictures of gene guns. So in this process, DNA coated on the microscopic pellets or a gold or a tungsten. Okay. Then it is having high velocity into the target cells. They are transferred with this type of gene gun. Then direct DNA injection and electroporation. Electroporation, it is the method of electrical impulse induce the transient or temporary pores. Here temporarily you can develop pores, and this is how you can insert your DNA so it can pass through the membrane. This is how it is. Here you have developed pores so you can pass. See, because of electric pulse, their pores are developed, and this is how your DNA can be transferred. Gel electrophoresis, it is the technique for separation of your desired fragment of DNA. Desired fragment of DNA separation technique. Okay, separate fragments of DNA after cutting off from the restriction enzyme. This is how these bands are formed. This is how these bands are formed. Look, DNA are poured. I mean, your culture, your sample is poured in this well, and then they are run in presence of electricity. Okay, into the poles. So DNA will start running in presence of this electric poles. DNA is negatively charged, so it will start running in towards the anode. Okay, and this is how shorter fragment will run faster, and you will get these type of bands. This is the complete picture of how DNA gel electrophoresis is done. Okay, look, after some time you are getting your DNA, still it is running. Okay, it will run, and after that it will form bands. Okay, on the basis of bands, where your desired DNA, what is the size of your desired DNA, you will get a few bands, and you can select that particular DNA. Process of recombinant DNA technology involves 5 steps: isolation of genetic material, cutting of DNA, gene amplification by PCR. That means you are amplifying your desired DNA. Preparation and insertion of recombinant DNA into host cell, obtaining the foreign gene product, and downstream processes. As you are doing it, you need to do it in bioreactors type of thing. So it will be require downstream processes. It is known as downstream process. So isolation of genetic material isolation you can do, and after that you need to cut it down, cutting of DNA. Okay, then gene is amplified by PCR. PCR involves denaturation, primer annealing, extension, and finally there is amplified DNA. Okay, so application of PCR involves diagnosis of pathogen, diagnosis of specific mutation, DNA fingerprinting, then detection of specific microorganism in parental diagnosis, and diagnosis of plant pathogen, gene therapy. Lot of applications of PCR. Then preparation and insertion of recombinant DNA into host cell, into the organism. Then obtaining the foreign gene product. Gene of your desired thing. Okay, desired phenotype that you need to obtain. And finally, it's downstream process, what you need to do. Okay. So process of recombinant DNA technology. In last, it is downstream process. After completion of biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. So process includes separation and purification, which are collectively referred as downstream processes. So product, you need to add some preservatives to sustain them, okay, to increase their shelf life. Such formation has to undergo through a clinical trials also, because I mean, if your recombinant DNA is successful, if your rDNA technology, your genetic engineering is successful or not. Downstream processes and quality control testing vary from the product to product. Again, I am insisting all of you enroll right now. Contact on these numbers. Okay. Next section of question answer, that is first question. It is rising of dough. Very simple question. Rising of dough is due to what? In formation of bread. Okay. In formation of bread, in fermentation process, this rising due to what? Dough is going to be doubled because of production of CO2, production of carbon dioxide. Okay, multiplication of yeast, production of carbon dioxide, emulsification, hydrolysis of wheat flour, starch into sugar. Okay, so dough is rise because production of multiplication of yeast and then production of carbon dioxide. So this both of the answer. So first it is because of multiplication of yeast. Then yeast is multiplying, so growth of that, it will produce carbon dioxide. So an enzyme catalyzing the removal of nucleotides from the ends of the DNA. Ends, look at the word here, ends of the DNA. It is exonucleases. We have discussed this in the class. The transfer of genetic material from one bacterium to another, one bacterium to the another bacterium. Genetic material from one to another through a mediation of a vector like viruses is termed as mediator. Here vectors here, they are viruses. Through viruses, it is known as what? Transduction. It is also known as sexduction. Okay. Which of the given statement is correct in the content of the observing DNA separated by the agarose gel electrophoresis? DNA can be seen in visible light. No. DNA can be seen without staining in visible light. No. Etherium bromide stained DNA can be seen in visible light. Etherium bromide stained DNA can be seen under UV light only. So correct answer is fourth D. Restriction. Restriction in restriction enzyme refers to what? In restriction enzyme, it is refers to, this word refers to what? This refers to the cutting of DNA at specific position only. Which of the following is not required in the preparation of recombinant DNA molecules? Which of the following is not required? Restriction endonuclease, ligase, DNA fragments, and E. coli. What is not required? E. coli is not required here. In agarose gel electrophoresis, DNA molecules are separated on the basis of their charge only, size only, charge to the size ratio, or all of these. Which of this is correct option? Correct option here, it is 7th B, size only. 1 second, is size only. Sorry. Its size. Okay. Then the most important feature in the plasmid to be used as a vector, it is origin of replication, presence of selectable marker, presence of site of restriction endonuclease, or its size. Which is most important feature of plasmid which is required? This origin of replication. Most all these things is required B and C, but most important it is ORI. While isolating DNA from the bacteria, one second. Which of the following enzymes is not used? Lysozyme, ribonuclease, deoxyribonuclease, and proteases. So deoxyribonuclease, which of the following has popularized the PCR polymerase chain reaction? Easy availability of DNA template, availability of synthetic primers, availability of cheap deoxyribonucleotides, availability of thermostable DNA polymerase. This is true, correct answer. Okay, thank you very much all of you for joining this class. Thank you all of you.